fourth session for BGA24. This session is on T2T assemblies with Verko by Sergey Koren. If you haven't seen the links in the chat already, then please have a look for them. And yeah, please take it away.
Yeah.
Stop sharing first.
Yeah. So I'm planning to start with a Git pod, and then while some of the commands run, because it can take a couple of minutes, we can go through a little bit of a background and overview. So hopefully you see my screen. This one here is what you get when you launch the Git pod. So if you're on the T2T page, make sure you launch the Git pod, and then the detailed tutorial steps are down here. So I want to give a minute for people to get their Git pod launched. It takes a minute because it's going to install Verko and do everything like that. But I'm going to start showing the commands, and I'm just going to be following along this hands-on. So if you ever want to go back and view the recording and you're not following along live, you can all the commands and everything I run will be available here. So you can just redo it at your own time if you don't have time to do something or you miss a part, you want to go back and redo it. So the first thing I'm going to do is just run Verko to get help. And this is a pretty good start to see all of the options that Verko has. Generally, everything after this advanced bit you can ignore unless you really know the details of the algorithms. And really the most important part are kind of the top three. So you were supposed to provide where you wanted to generate the assembly, your hi-fi, high accuracy data. So this can be BagBio hi-fi. This can also be ONT duplex data or ONT Dorado or Hero corrected data as well. And then nano are the nanopore data. The longer the better. We typically recommend having a good amount of coverage in the ultra long data of reads greater than 100 kilobase, because this is really key to resolving some of the complex repeats in the genome. In terms of other parameters that are useful is you can if you have high C data for phasing. So Verko is really designed to have some kind of phasing information. If you don't have something like a trio, high C, strand seq, pore C, all it's going to do is give you essentially unitigs for the entire genome. So it's going to be twice the size of a genome you expect if it's a diploid genome, or 4 times if it's tetraploid, and so on. So if you have high C data, you can provide high C data with these options. If you have ONT pore C data, you can provide it. Or if you have trio, you can provide databases for the paternal and maternal haplotypes. And all this is also documented on the Verko GitHub page. So that's kind of the overview of the options. The most important options, like I said, are the input files, the high C or trio or pore C phasing. And then the only other option that you might want to use is this screen option, which allows you to screen out either common human contamination or human cell line. So this will find all the rDNA sequences in the human, the mitochondrial sequences, and the EBV in the case where commonly human cell lines are EBV transformed. So this will get rid of those things so that your main assembly FASTA that you then submit to NCBI or wherever won't have those sequences there because NCBI wants you to separate the mitochondria out. So this kind of just does it for you so you don't have to worry about it. Yeah, that's kind of the main option. So I just want to make sure people are able to start Git pod and start following along. Hopefully so. If you don't, either message on Discord or in the chat on the Slack, on the Zoom, but I'm going to start going through the commands here. So we've got some test data that I happen to download onto the Git pod here. It's also available through this link that's posted here. And so what this is going to run, so we can go through, so we're going to output all our assembly into the folder named test. We have some hi-fi data, some ONT data, and we're going to be using high C data for phasing. So we've got our Illumina pairs, pairs 1 and pairs 2. And I'm also going to screen common human contaminants. And I have this extra option called dry run, which I'm going to show in a minute what it does. So when I run this, it's going to tell you everything it plans to run. So this is a summary of everything that's going to run. And then down here, it's going to do a step-by-step overview of what Verko is doing. So this is not actually running anything. It's just telling you what it plans to do. And so this can be useful if you, for example, have an assembly and you interrupted it for some reason, or there was an issue on your cluster and you had to kill it and restart it. And you want to make sure it's not going to rerun the same things that you've already done. This dry run could be just a good, quick, useful start to let you know what it's planning to do, and it's not planning to rerun anything that you don't expect it to. And so you go through this. If you go through these steps, it's kind of the rules somewhat explain what it's going to do. So it processes the hi-fi FASTA first. It partitions your Hi-C data and your ONT data into smaller batches. It builds the graph for the assembly. So this is the multiplex, the brown graph that it builds from the hi-fi data, simplifies that graph, processes the ONT data. There's alignment steps in between here where it's aligning and combining the ONT data, simplifies that graph after the ONT data, then removes bubbles. And then lastly, it generates the consensus. So here it's generating the final sequences. And then the last bits down here are all related to Hi-C phasing. You can see then it prepares scaffolding, et cetera, and does the Hi-C. And then at the end here, it says, as you said, this is dry run. It didn't actually run anything. It just told you what to do. So this is, like I said, useful, and it gives you for all of these a reason for why it's running something. So in these cases, it's saying it's missing files from the previous step, which is not surprising. We've never run this. Obviously, they're going to be missing. But if you see things that it's saying that the files are out of date, or it's saying it's forced execution or something unexpected, that might indicate that, if you had, like I said, interrupted an assembly and moved it around from different folders, it could confuse snake-make. And so this is like a good sanity check. So we're going to run this assembly. So this is the command now for real. This is the same thing. You notice I've removed the dry run option, and I've added the no correction option. And this is only for this tutorial. This means don't correct the hi-fi data or whatever that input high accuracy data is, whether that duplex, hero, etc. The only reason I'm doing it in this tutorial is because I want to run it faster. And so this saves compute time. But in general practice, you probably would not do this because you want to correct the data. It gives you a better assembly. And it's not worth saving the little bit of compute time for it in a normal situation. So I'm going to let this start running. And so now you can see for now, rather than listing all those jobs immediately, now, you can see that it's started some and it's paused now because now it's actually doing the computation. And for each job, it's telling you how many CPUs and memory it plans to use. And if this was on a cluster, it would submit those to the cluster and wait for them to finish. In this case, they're all running on this grid, on this Git pod on a single instance. So while this is running, I'm going to go through a little bit of a kind of background overview of Verko, just to give you some more background, and then we'll go back and start looking at the actual outputs of the assembly. What do you do when you have an assembly, kind of, and some of the most basic kind of troubleshooting tips for generating a Verko assembly. Right, so the motivation behind Verko is really to generate telomere to telomere assemblies for all genomes. So you probably know that we recently finished this human genome, and this was a very manual effort. And it was really enabled both by new algorithm development, but also by this combination of very high accuracy at the time hi-fi data with very long, ultra long ONT data. And why are these two data types so useful and so complementary? First, if you're not familiar, nanopore data is essentially an electrical-based system where you have this membrane, you have a pore that's embedded in the membrane. There's a motor protein on the top here that pushes the DNA through at a fixed rate. And then you get this kind of squiggle traceout that measures essentially the flow of the through the pore, and the DNA impedes that, so you can see, depending on what base is in the pore, that squiggle changes, and then you use machine learning to translate those squiggles into AC, Gs, and Ts. So these have the advantage that they can be extremely long. It's a very portable instrument too. Here you can see our collaborator Matt Luce sequencing in his hotel room at AGBT a few years ago. So it's very portable. They could produce very long sequences, but it has lower base quality. So the ONT has been steadily improving this. So essentially, it seems like whatever DNA you can get to the pore, it'll read. PacBio sequencing, PacBio has been around for a while, but the CCS is somewhat newer. And if you're not familiar with PacBio sequencing, the way it works is you start with this double-stranded DNA. You attach these kind of dumbbell adapters on it, except they call them smart bell because they can't name them dumbbells, right? And so then you load these into these small zero-mode waveguide wells in this plate, and there's a polymerase there that starts reading this sequence and synthesizing the other strand. And because you have these adapters that are circles on the end, you never stop reading. So the polymerase is just going to go along, and once it finishes reading one strand, it's just going to go around the loop adapter and start reading the other strand, right? And so you're going to get these kind of passes where you start reading somewhere, you read a bunch of this strand, you go through the adapter, read the other strand, go through the adapter, read the same strand again, through the adapter, read this strand again, right? And so you're going to keep reading forward, reverse. And even though each one of these times you read it, it has, about a 15% error rate, then when you pile them all up on top of each other, because the errors are all random, the final consensus of that is going to be very, very high accuracy. And so it's shorter than the ONT data because you have to be able to read a molecule multiple times. So it can't be more than about 20, 25 kb because the polymerase just can't read a longer molecule enough times to get a good consensus. But they are extremely accurate, near perfect reads. And to kind of give you intuition for why you want these kind of types of reads and why the two of them are complementary, you can think about how an assembler will view these reads, right? So let's say we have this toy genome, and we have a repeat with two copies where one copy has these two red SNPs that differentiate the two copies, right? And so if we sequence with ultra-long data here, that the little dashed lines indicate sequencing errors in the reads. If we keep reading this enough and get enough coverage, we're going to get these purple reads, which are long enough that they could be uniquely anchored outside of the repeat, right? So even though we never actually had these reads don't actually accurately capture the SNP variants because they have higher error, we still know that this purple read has to go to this purple read. It can't go over here because this bit between the repeats is unique. And so this sequence is going to look different than this sequence, right? And so while we can't tell if this read should be attached, over here or over here, we can't tell that this read goes to this read, goes to this read. And so we can resolve this genome, even though we never actually accurately captured each individual SNP in the repeat that differentiates them. And this is pretty intuitive. This is kind of how you think of assembly, like longer reads better. Yes, this all makes sense. But accuracy also matters. So let's say now if we're sequencing the same kind of genome with hi-fi reads, now none of them are going to be long enough to span a repeat like I showed you in that picture because the repeat is too long. But if we sequence it again, we again can resolve this repeat because when we look at all these reads that have piled up, we know that this purple read has to go to, the purple read here to here, and we can't go from this purple read to any of the blue ones because all these reads have this consistent variant that we're seeing. And so it doesn't make sense to connect a read with a variant to a read that doesn't have that variant, right? And so you can imagine that the more accurate the reads are, the better you'll be able to do this kind of resolution. So even though none of these reads actually span the entire either repeat copy, we can still accurately assemble this genome as long as we can be confident that when we see this kind of red mark in a read, it's a real difference in our genome and not just the sequencing error that the instrument put in all the reads, right? And so this, I think, is less intuitive maybe the first time you hear it, but the more accurate the reads are, you can resolve actually, as long as there's variation in your repeats and they're not identical repeat copies, you can resolve them even though you never span any of those repeats, right? And so, to get the best of both worlds, because there are actually in the genome, there are both repeats that are really long and identical, and then there are repeats that are that are extremely long, like megabases long, like centromeres, but they have enough variation that we can actually tile across them with something like hi-fi reads. So for this project, we did, for the T2T human project, we built an hi-fi accurate assembly graph, and then we layered the nanopore data on top of this to resolve this, right? And so we did this by hand, and I'm going to show you a couple of examples of how you do that. So here's an example of a graph. If you've never looked at this before, the long bits here are sequences, and the edges here are connections. So it says that this node shares some sequence with this node. It also shares this sequence with this node, and this node shares sequence with this node, right, and so on. And so if you look at this kind of graph, there's a repeat here, but if you stare at it long enough and hard enough, you can find a traversal. If you assume that every node has to be used once, there's only one way to traverse and use every node here, because if you start going, for example, this way, then you have no way, you'd have to go backwards to use this node. You can't do that. So the only way to walk through here is to go like this, as the path shows. And so you could resolve this without even having ONT data, just by using kind of graph theory and rules about assumptions of the number of times you have to traverse a node. And then there's only one traversal that satisfies that criteria. In some cases, though, you can't decide how to traverse it no matter how hard you look at it. And so this is another example. And the problem here is that we have to go through this node twice. And the problem is we don't know, should we go this node, and this node first, or should we go this node, and this node first? And these nodes are too big for the hi-fi data to be resolved. They're too similar. And so we need a longer ONT read to align through it. And in this case, the ONT reads can tell us which way to go. And this is another example that's also looks pretty simple, but the problem here is that we don't know whether we need to read this node in this direction or this direction, right? So because when we go, we know we must go in here, and we have to use this twice. But from here, we can go either in this direction and back out, or we can go in this direction and back out. And so even though it looks pretty simple from a structural perspective, we still need the ONT data or any kind of longer data to tell us which direction we need to traverse this. And in both cases, the ONT reads can align and tell us, how do you actually traverse this? And so this can be done, as I just showed you, right? That's just 3 examples. We just resolved them here. But obviously, that's very time consuming. So you get a graph that looks like this, and then you have to turn it into something like this, where you've figured out what all the chromosomes are. You've assigned bits that there are some regions that the hi-fi reads can't sequence in a genome, where they just drop out sequence. So you have gaps. So you have to figure out that like this little bit of a chromosome is connected to another bit where there was a gap, and you have to figure out that all these components are part of the same chromosome. You have to look at all those little tangles and figure out how the ONT reads align through them. And so this is very time consuming to do, which is why it took us so long to finish that human genome. And it's error prone. I'm not going to spend too much time explaining in detail. This is a strand-seq validation. And so what strand-seq is showing you is the blue indicates that everything is in one strand. And then this little bit of orange says the strand switches, and then it goes back to the same green strand, right? So what this is saying is there's an inversion centered around here, where the manual assembly inadvertently flipped part of the genome. And this is a real example from chromosome Y in the human genome. And the node that got flipped is one of these nodes in the middle of this big tangle that we were trying to manually resolve. And you can see that when it was easy to do it when I showed you just a couple of nodes, but when you've got these long paths that are extremely similar, it's really easy for a human to make a mistake. And so we wanted to automate this and stop doing this manually. And so that's where Verko comes in. And so the idea with Verko is that it will do all those steps that I just outlined for you, but it'll do it for you in a consistent way. It will make those mistakes that a human makes when humans can't, keep all this in their head. You make a mistake when you're doing the traversal. You don't have to make manual decisions about the ONT resolution, et cetera, et cetera. So our kind of recommended recipe, and this is per haplotype, so double these numbers for diploid, quadruple if you have more polyploid, et cetera, et cetera. So we recommend about 20 to 25-fold some high accuracy coverage. This could be hi-fi, duplex, hero data. 15 to 20x of the greater than 100 KB reads, and then either Trio or Hi-C. And Verko is available in Conda. We just had another release today, so you can install it on your system easily, and it's compatible with most grid systems as well. And it does exactly what I just told you we do, right? So first it takes the reads and homopolymer compresses them and corrects them. This is because most of the errors that hi-fi reads make are actually homopolymer counts. And so by compressing them, we just get rid of most of the errors that are left in the reads. We build this multiplex, the Brown graph, which is built using only the high accuracy data. And that just shows you a kind of pictorial representation of it, where you've got this graph. Then we layer the ONT reads on top. And so in cases like this, where we didn't know how to, if we need to connect this node and this node, or this node and this node, the ONT reads can tell us the unique resolution. And so we can now merge these blue nodes, merge these orange nodes with this, duplicating this node in the middle. And we can figure out how to traverse these kind of repeat tangles based on the ONT alignments. And so you get this kind of simplified graph. This is all done automatically for you. And then whatever phasing information you have, whether it's trio or not, so you need to know whether it goes green, gray, blue, gray, purple, or green, gray, orange, gray, purple, or orange, brown. Like you need some kind of other information, and that's where the Hi-C or the trio comes in. And so in this case, if we have trio where the maternal haplotype is colored red and the paternal is colored blue, this tells us, okay, so we should follow this path here. So that would be the green, gray, blue, gray, purple. And for the bottom one, we follow this path. And now you've got your two haplotypes, right? And so at the very end, you get your final consensus for both of these haplotypes. So you may be asking yourself, okay, that's great. You try to automate it. And so how well does it do? And so with the latest version of the assembler running on about 100 human samples from the HPRC, the Human Pangenome Reference Consortium, which average about 60-fold hi-fi coverage, 30-fold of reads greater than 100 KB, and about 70x Hi-C. So you can see this is pretty much following the recipe I described for a diploid genome. We get 40 out of 46 human chromosomes as T2T scaffolds. So this means they have telomere on one and telomere on the other. They may have one or two gaps in the middle, but they're essentially being resolved across the entire length. And if you know, in the human genome, there are 5 acrocentric chromosomes. So really, you can't get more than about 36 T2T contigs. So 40 T2T scaffolds is pretty good. That means we're resolving at least some of these acrocentric chromosomes as well now. And just to give you an idea of memory and runtime for Verko, so this is a bunch of those samples. The runtime, this is the median. It's around 2,200 CPU hours and less than 100 gigs or about 100 gigs of memory for most of the samples. There are a couple of outliers, one way or the other, that had maybe higher coverage or something else, or more complex genome structure. But for the most part, a human genome with Verko takes about 2,200 CPU hours and about 75 or 100 gigs of memory. And it mostly the runtime depends on having how much ONT data you have, because the alignment of the ONT data is one of the most expensive steps. And so you can see there's a slight trend, though it's not very strong with the ONT coverage. And we can look at this data another way. This is the same kind of thing that I showed you before, except now we're grouping our results chromosome by chromosome. And so the bits in blue are T2T contigs, and the bits in red are, the T2T scaffolds on top of that. So you can see that about half of them are T2T contigs. So that means it has telomeres on both sides and no gaps. And are automatically resolved from the assembler with no manual intervention. And then about 90% are T2T scaffolds. And it varies chromosome by chromosome. So I mentioned the acrocentrics. These, if you're not familiar, are the chromosomes in the human genome that contain the rDNA arrays. These are very repetitive. And so we can't resolve the actual sequence of the rDNA arrays at the base level yet, which is why there's almost no T2T contigs, except for maybe cases where one of the chromosomes might have a very short array, in which case we have ONT reads that tell us how to resolve it, which happens sometimes, but it's not very common. But we can get T2T scaffolds for, about 75 or so percent of them, where we can still tell you, even though we can't tell you the base level resolution of the rDNA, we can jump across it and say, here's the Q arm of the chromosome, here's the P arm of the chromosome, and the only bit we couldn't figure out in the middle is the exact base level sequence of that rDNA array. And that's because the rDNA array is a 50,000 base repeat that's in tandem over and over and over again repeated. So it's very difficult to resolve, even if you have ONT data. It's still not long enough because these are megabase long sequences, arrays. And most of the chromosomes look pretty good, but there are some outliers that you may notice other than the acrocentrics. So the acrocentrics stand out because they are only scaffolds, and that's because of the rDNA array. You can see that the X and the Y chromosome work pretty well, as you get almost half of the Y chromosomes resolved T2T as contigs also automatically, which is better than we had hoped when we started this, because that is also a very repetitive chromosome. It has a lot of large satellite arrays. So these are the acrocentrics, but then you see some, like chromosome 9 here looks kind of off, right? All the other chromosomes are peaking up there other than the acrocentrics, but then 9 looks almost as bad or even lower resolved than the Y chromosome, which we wouldn't expect. And if you look at all of the chromosome 9, for these 101 samples that we assembled, the dark blue means that it's a T2T contig. So each row here is a genome. And dark blue means it's a T2T contig, light blue means it's a T2T scaffold, and white means not resolved. And so you can see that what happened is it was doing just fine, everything was being resolved. And at one point it just stopped being resolved, and there's very few of them being resolved where everything's white. And so this coincided, we found, with a switch to the new PacBio sequencing instrument, the Revio, which switched to a shorter movie time. And as a result of that, it didn't have coverage in one of the repeats in the chromosome 9, near the chromosome 9 centromere. And so as a result, all these chromosome 9s were not resolved because they were split into two components that we couldn't reconnect because the repeat was too complex to resolve with just ONT data alone and no hi-fi coverage at all. And the good news is that if you extend the movie time from the 24 hours to the 30 hours, it seems like this is mostly addressed and the hi-fi coverage comes back. But that's just to give you an example that, at this point, because the assemblies have gotten so reliable, when you're assembling the same genome like this over and over again, when we see something going off or a problem in the assembly, it's usually not because of the assembler. It's actually a lot of the time is a problem with the data, like this. So that's all great, but you may ask, you claim that it's T2T, but how do you know, right? And so there's kind of standard T2T metrics QC metrics we run. Obviously, one is the count of these T2T contigs and scaffolds. We typically measure QV. This is the remaining errors in the assembly, essentially any base in the assembly that's not present in the sequencing data. And this can be done with tools like Mercury and Yak. We measure Hamming and switch error rate. So this is how frequently you mix up the haplotypes if you have trio data available. So if you, for example, have trio data, but you assemble only with high C, then that's an independent validation that your high C data correctly phase the genome and you don't mix maternal and paternal haplotypes all over the place. We also rely on BOOSCO or CompleteSM, which I know there's a session on later in this BGA workshop, which identifies missing and duplicated core genes. And so this gives you an idea of if your genome is actually complete or if it's missing. It also is useful for detecting misphasing, because if you take a sequence from the maternal haplotype and mix it up and put it into the paternal, now the paternal genome is going to have duplication and the maternal one is going to have missing genes. So it's useful to look at that also to measure Hamming and switch error rate, especially when you don't have a trio available to validate independently. There's the non-T2T continuity metrics. And then there are several tools that use alignment-based evaluation with the idea that if your assembly is the genome that was sequenced, then the reads should match it. And so if we map the reads back to the assembly, they should agree with the assembly. And if these tools flag regions where they don't, where there's disagreement of the reads with the assembly. But some of the things don't actually get captured by this kind of thing. So if you imagine if we make this kind of scaffold and we make a huge gap and there's a bunch of sequences we just didn't place. These should have gone in the middle here, but we just left them all alone as their own individual contigs. The QV is going to look fine. The missing genes are going to be fine because if there's any genes in here, they're still there. And the alignment is also going to be fine because these bits are correctly assembled. But really, we should have put these sequences into this gap, right? So there's not a there's still bits that this kind of QC misses. And so while these QC, so really what I want to say is that these kind of QC metrics are just the bare minimum. So you should run all these kind of tools and evaluate them. But just because those don't show a problem doesn't mean your assembly is perfect. There's still ways to have these kind of edge cases. And so you still want to make sure and sanity check the assembly. You want to look at the assembly graph, which I'm going to go through at the end after these slides when we go back to the command line. You want to make sure that there's not sequences that should have been placed where they weren't, and so on, and just kind of spot check and sanity check the assembly afterwards. Don't assume just because these kind of standard QC metrics don't indicate a problem, there isn't one. And the nice thing about some of these metrics is people use them for a single number, right? So you run the QV metric and you say my QV is Q60. But if you actually look at the Mercury output, it will actually tell you the exact coordinates where the errors are. And it'll also give you a track you can load in IGV to see those errors. And so if you see that there's a whole bunch of errors concentrated in one area, that could indicate some kind of misassembly and not necessarily a base problem, right? Error. The same thing with the YAC trio eval, which gives you where the location of the switches are. So is it a lot of switches all over the place, or is there just one big switch where we took a big contig and switched it between haplotypes? It kind of can give you a better sense of what actually is going on in the assembly other than just one big summary metric. Again, for the CompliAssemble and Busco, it tells you where the gene is that's missing. So you can look for that missing gene in the other haplotype. And together with the Verko outputs, you can actually localize that to the assembly graph and see what's going on in the assembly a lot of the times. And I'm going to show the assembly graph and how you use this to move between the final sequences and the assembly graph using those files. So I want to wrap this up by going through a few genome, if there's one thing you take out of this, tutorial, it's to trust and love the genome assembly graph as much as I do. The first thing we do whenever we run a Verko assembly usually is just open the graph that it outputs because it is very informative and it kind of gives you an idea of what you're working with. So I'm going to go through a few examples of graphs, genome assembly graphs. These are a mix of human and non-human. So this is a human assembly.
Sorry, Sergey, can I just butt in very quickly? Apologies. Yeah. Can everyone click around in the Git pod quickly, just because it has a tendency to go to sleep after 20 minutes?
Right, let me do that too.
And if that happens, you have to rerun everything.
Which is annoying. Yeah.
Pretty annoying, yeah.
Yeah.
So that's just your 20-minute warning.
Let me do this then. Let's just go back to Gitpod and I'll resume this. I'll finish it up at the end of the session because I'm pretty sure it finished running. Mine finished running. And I'll do the kind of simpler graphs there, and then I'll go through the other graphs I was going to show at the end of the talk. So let's do it that way. Because I didn't think about it timing out, but thanks for the heads up. Right, so now that we've generated the assembly, we can now see in your folder here, the test output. We've generated the assembly. There's intermediate steps of all that I just went through in the slides, right, where there's the correction step, there's graph building, there's the aligning the ONT data. using the ONT data to resolve repeats, finally generating the consensus, and then high C phasing. And in the documentation here, description, I listed kind of the summary of what the sequences are. So assembly.fasta is kind of your main output. This is your assembly. This includes both haplotypes and anything that could not be assigned to a haplotype. In some cases, usually there's not too much unassigned sequence, but it is always worth looking at it, especially, for example, the sex chromosomes sometimes can end up as unassigned, especially when there's not just a simple XY kind of, or things that we haven't seen before in new species that we haven't assembled previously and didn't expect their graph structure or their genome structure. Then each haplotype, if you specified phasing information, you're going to get a haplotype 1, haplotype 2. And then this GFA these two GFA files are the graphs that we're going to look at. And then there's since I had the screen option, you see detected a mitochondria or tried to detect mitochondria, tried to detect rDNA, and tried to detect EBV. In this case, my test data set didn't have those, so these are empty. If I do that, you can see that they're empty. But if you had a full human data set or another data set, they would presumably find and identify these sequences. So we can look at these in detail. So if I do this, we can see we have two sequences. Both are about 4 megabases in length. One in each haplotype. There's nothing unassigned in this assembly. Everything is either haplotype 1 or haplotype 2, and it's about a 4 megabase sequence. And so you may, I mentioned that we can go from these sequences to actually where they are located in the graph, what the assembly saw. And so if we look at this file called SCF map, it's going to say, okay, well, so haplotype 101 corresponds to this path. Haplotype 202 corresponds to this path. So if we want to find this path, we can just grep that sequence in our path file, which is right here. And this only exists if you have a trio or high C data. If it's not phased, your output is just going to be, rather than having a path like this, it's just going to say this haplotype 101 is, let's say, UTIG42, like a single node. But so what this says is that this path for this haplotype, you first take this node in reverse. That's what the minus means. Then this node in the plus strand, and then this node in the plus strand. And you can do the same thing for the other haplotype. And you see, then it's got its own nodes again, reverse, forward. So we can now take a look at this graph and see what this looks like, right? So if you do this, it opens Bandage and it opens it in this other window that hopefully you have, because you have to have pop-ups disabled for it to open. And so in Bandage, we can load the graph here. If I go to workspace and then test, and then the assembly, it doesn't matter which one you load. We usually load the no-seq because that is much smaller. Essentially, all the sequences are just, the nodes just have length, not actual bases. And when you're working in Bandage, usually it doesn't, you're not using the sequence of the nodes. You're just viewing at the graph structure. And so you don't need to spend all the time loading the sequences. So we typically open the no-seq. And so if I open this and I draw this graph, we get this nice picture of the graph where each one of these long bits is a sequence. So you can see their names when you click on them. UTIG41, UTIG42, UTIG3. You can select multiple ones. And we can also load this CSV file that Verko again automatically produces for the colors. And that's going to tell us which nodes. So this is the maternal haplotype, and this is the paternal haplotype, right? So it gave us the coloring for the nodes here that it derived. One useful thing to do in Bandage, and I know there's a whole Bandage NG tutorial, but one thing I usually do, and I wish this was the Bandage default, is to turn on this arrowheads in single node style and the graph appearance to on. And so the reason you do this is, so when I look at this node, remember I told you we took one node in the forward strand, another node in the reverse strand. But when you look at this node, it's like a square block. You can't tell where's the start of it. What does it mean to take this node in the forward strand, the reverse strand, right? And so when you turn that option on and you draw the graph again, now you can see that the nodes have little, like a little arrow like this, you can see here. And it has a little indentation on the other side, right? So like over here, you can see where it's indented and then it has the arrow. So if you take this node in the reverse strand, it means you would start from this pointy end and go in this direction. If you take this node in the forward direction, it means you start from this little indentation. And so it just makes it much more useful to be able to see what the node looks like. So let's see what. And you can show information like the name of the nodes. Let me make the font bigger. Too big. But now you can label the information for the nodes, so you can see if I move them around a little bit. You can see that the names of the nodes now, their coverage. So this is about 20-fold coverage per haplotype, which is what we expected. And consistent, you can see the coverage is consistent across the haplotype, and you can see the names. And so now if we go back to this other path that we were looking at, you can see that we had one of our paths was you take 4.2 in reverse, 4.0, 4.5. So if we go back here, we have this node. I'm going to make the font slightly smaller so that it's not so big. So we took this node 4.5, right? So I'm going to move it over so it's a little bit easier to see. So we started with 4.2, 4.0, 4.5. So 4.2 is this one over here, which we took in the reverse, which you can see means we started from the little arrow bit. So we started from over here and we went in this direction. Then we followed this edge and we went to 4.0, right? And then we followed this node in the forward direction. And then we went to follow this edge into 4.5. And so that represents our maternal haplotype, right? And then for the other sequence, we took 4.1 again in the reverse strand, which means we started from here to here. We followed this edge, took this sequence 4.3, and then the sequence 4.4. Right? And so if you don't want to do this again, you can look at this graph online here. I have the picture saved. You can look at it and see the same kind of information that I'm showing you there. So another kind of common thing we do besides, so this graph is useful, but it's not, it doesn't really tell you anything about what the sequences are, right? Like I have no idea what this chromosome is. I have no idea what the species is. Well, I know it's human, I guess, but I don't know like what these chromosomes, where this is in the genome. I don't really know if there's telomere anywhere in here, if I'm at the end of the chromosome or not, et cetera, et cetera. And so it's useful to have a few scripts that we have that are in this Git pod, but they're also available at this link that I posted in GitHub. And so one useful script that is also one of our common PC things is to run this T2T script. And so this just counts nodes that have telomeres on both ends. And so in this case, it produces a file called telomere. And so this tells us that these two haplotypes have telomere at the start. So if we look at this sequence, that means that these gray nodes we've been looking at, right, 4.2 and 4.1, these have telomere at their ends. And that makes sense because that's where we start from. And we know from the coordinates here that the start of that sequence has a telomere. the first 4,000 here and the first 7,000 here. Right. But we also have some convenient scripts which will add this information into the graph. So you don't actually have to know this from the start. So now it produces a graph. I did it zoom in and not let me zoom out. So it produces this new graph that has telo in the name, so that adds a telomere. So if I open this graph, and now I'm gonna load the same CSV file for the colors. The graph. And so now you can see that it's added these little green nodes over here that are named telomere that's attached to this node, and then telomere attached to this node. And so that makes it, if obviously in this graph, it's not that interesting because we've only got a few nodes because I took a small sequence to run for this tutorial. But if you're looking at a new species, especially you might not know how many chromosomes it has, you're trying to see in your graph how many components are actually complete and have telomeres on both ends. This can be really useful to see because you'll see that, okay, this component can't be a whole chromosome because it's only got telomeres on one side over here, but then there's no telomeres on this side over here. So we must have another bit of that chromosome somewhere else in the graph, right? So if you saw this, you would know you have to have at least two complete coverage breaks in this graph, presumably because the rest of the chromosome is somewhere else that we don't see, right? So this is a very useful way to look at this graph. And like I said, these scripts are all available in this GitHub I linked. And we also have a quick helper script that will align the graph to the reference. And again, I can run this. And so so if you look, the last one I ran was just get T2T, and it just takes the assembly fast. It relies on seq_tk to find the telomere. And it'll just, take the sequence fast, find all the gaps in it, find all the telomeres in it, and make you a summary. This one will run mashmap to align to this reference. This is the identity. So if you don't specify the identity, it'll default to 99, but you can specify an identity and then what you want to align. So this is reference, identity, what you're aligning. And this one takes a little bit longer, but you can see it's done. And this produces kind of two useful outputs. First, it tells you a summary of the haplotypes to what they got mapped to. So in this case, this sequence got mapped to chromosome 12. It's 4 megabases. Chromosome 12 is actually 133 megabases in the reference. So we can see this is a tiny fraction of that chromosome. So we're missing a lot of sequence. And the same thing, this is also chromosome 12, which makes sense. It's two haplotypes of that same chromosome. And we can actually look at the output, and we can see that the sequence aligns full length to chromosome 12, and it aligns to the very end of the chromosome. So that explains why it has the telomere there, because it's the very end of chromosome 12 on the Q arm where it has the telomere there. So that's the very end of chromosome 12. This also tells us that our assembly is essentially reversed with respect to the reference, right? Because remember, we started with the telomere nodes. And so we're walking from the telomere of the Q arm and towards the P arm of the chromosome. So if you wanted to make this match the reference, you'd want to flip this assembly reverse complement. So let me go back. So let me see, are there any questions? I want to pause and see, are there questions about that output or kind of things we want to do with the graphs? Or any other questions from Bandage, Verko output, before I go back and finish looking at some of the slides. To answer the question in the chat, yes, the ONT gets mapped to the assembly graph with a graph aligner. So one of the steps in Verko in this, if you look at in here is there's alignments. So this is actually, you can look at all the scripts that Verko runs. And so you can see under the hood, what it's doing is running Graph Aligner to align it to this intermediate graph that it's produced. And then it uses that information to simplify the graph. But yeah, the reads are aligned directly to the graph. In terms of the validation and the QV, in the QC, we usually align directly to the final assembly. Yeah, and you can see I've also posted that other graph that I was showing with the telomeres that we showed you. That is, you can view it again if you don't want to start the Gitbot. You can go through the tutorial and read it through. So there's a little bit of extra things that I was going to show you, which is, let's say you've mastered these graphs, you know what you want to do, and you decide you want to change the path that the assembler takes, which is possible to do, but I'm going to do that after I finish the slides, because I think it'll be a good breakup that we can do the 20 minutes, be without timing out, finish off the slides, see if there are questions about that, and then we can, I can quickly show some of that assembly editing. Yeah, so those were, I took a very small bit of chromosome 12, obviously the very end, because I wanted to show the telomere sequences. Obviously, real graphs have more than 4 nodes, or 6 nodes, but they're still very informative, and they're surprisingly easy to look at even for a full human genome. So here's an example. This is now the whole human genome. And so now that you kind of had a taste of what it looks like on a toy example, we can start looking and seeing the features we want to see in this graph and the features we don't. So this is good. We like to see this. There's a whole chromosome 18 from one haplotype. It's been completely phased out. There's, there's 2 telomeres on the ends, and there's really nothing for the assembler to have to even do with phasing, because it was already completely phased just with the data. The IC or the trio here was not necessary. It just added color, but we would have gotten this entire chromosome end to end, even if we didn't have high C or trio data. Right, and most of the cases where it's a single haplotype like the X or 4 here, we see the 2 telomeres. Then when there's 2 haplotypes mixed, which we can still resolve because they're colored, so we know that we need to take this blue path and this red path, we see 4 telomeres, which is good to see. We see an example of something we don't want to see here, which is, like I was saying, you've got 1 telomere or some sequence and then nothing. So this is a chromosome that is broken up, so the rest of it is somewhere else, because we don't see a telomere here. So we would need to find what happened to the rest of this chromosome 22. Right, and this is an acrocentric, so it should have attached to this rDNA cluster. And these are the acrocentrics. This is why they're difficult to resolve that I mentioned. This big black hole in the middle is the rDNA array that's just repeated over and over again. And it's extremely similar between all the chromosomes. And then you have the long arms of the chromosomes and then the short arms coming off here. And so we can tell you that this connects, for example, this bit connects to over here, but we can't fill in that black hole sequence at the base level, right? So yeah, so the things you kind of want to see, large nodes, there's not that much gray, which means there's not a lot of stuff that's homozygous. We've been able to phase everything out. Obviously, this is going to vary with your genome, heterozygosity level, with your read lengths, and also, the genome structure. Like you can see here, in this individual, chromosome 10 has more homozygous sequence in chromosome 20 than the other ones, which is why they have more gray nodes, but there are not that many of them relatively in this graph. You can see the telomeres again in the components for when we have the diploid 2 in the haploid components. And we don't really see a lot of hanging edges. I showed you the one in chromosome 22. But usually we end either at the telomere or we have a neighbor to go to, which is good to see. And there's this clear rDNA tangle, which we expect to see in human. Obviously, this is species specific. It's going to change if you're looking at not a human genome, right? This is more of an average graph now. This, has about 3 times as many nodes as the previous one did. It's a slightly worse assembly. It's still pretty good. I mean, it's got, 37 T2T scaffolds. So a little bit below our average, but not too far off. And so the things you can kind of see, there's a lot more gray nodes. This is this is because the reads were shorter in the initial graph, so there was less stuff that could be resolved, and there wasn't enough information to phase some of these homozygous regions, so you end up with more of them. There's places where, like this chromosome 17, we've got telomere here, telomere here, telomere here, but then we're missing a telomere over here, so that means we probably had a coverage dropout or sequence loss somewhere where we wanted to have it. Another example over here in chromosome 16 where we are missing, there's this dead end here instead of going to a telomere. Right, so that's hanging edges. Here again, we have chromosome 9 where we've got, it goes to over here, but then there's no telomere here, there's nothing, no neighbor node. And so something's gone wrong here. There must be more of chromosome 9 somewhere else, which was not connected, and it's actually down here. So this is an example of that issue I mentioned with the early 24-hour Revio movies where chromosome 9 had no coverage in the middle of it. And so it ends up as kind of this two components, one with telomeres and another with telomeres, but no way to connect the two. Right, so that's this example. And then kind of the more ugly graph. Now, in terms of the T2T stats, this one actually comes out okay. But the kind of things that are not so good in this graph is there's a lot of the gray nodes. There's also there's also some yellow nodes. Actually, I don't see yellow nodes. There's more gray nodes in this graph, so less phased. There's about twice as many nodes now as the previous graph. And there's this really complex, really homozygous node in chromosome 19 that if we dig into, there's a non-diploid structure here, which probably is pointing to a cell line issue or some kind of issue. And so what we can see is we expect to see this kind of structure like we see on chromosome 18, where we've got two haplotypes. You see they're going along here, 30 coverage for each, which is what we expect. I told you we average 60 coverage for the diploid. But then at some point we just transition and we end up having one haplotype that has about 40 to 50x coverage, and then the other one that has 10x. And so it looks like there's some kind of maybe like there's cells that have lost one of these chromosomes, something going on. Maybe it's not diploid anymore in the cell line. So this would, we would be suspicious of the cell line and want to dig into what's going on here before we use this assembly. But in this case, the graph looks bad, even though the T2T stats are okay, but it has a lot of nodes. And it's mostly just because of this one area where the cell line looks like it's abnormal. And so the assembler is pointing to an issue, not with the algorithm or the software or your data, but actually your sample. And so this can be useful to learn from your graph if you look at it and dig into it, right? And so this happens in the middle of the chromosome, and then it goes like this all the way to the very telomere that one haplotype has 10-fold coverage and the other one has like 45 or 50x. So to give you some idea of what can go wrong with Verko when you look at these graphs, you can get these very few T2T scaffolds or very fragmented graphs. So this example is actually from an example from the Darwin Tree of Life that was an ultra low input protocol, so it was amplified. And so because of the amplification, there's a lot of coverage dropout. So this graph is just very fragmented because there's a lot of regions of the genome that just don't have sequence. So if you see this graph, you know something is off, and you're not going to get a very good assembly from this kind of data set. If some regions that it can vary, I said some regions might be homozygous or phased or not, but actually it varies quite a lot between genomes, and it varies even within a genome. And so you can have a genome that has a lot of very nicely phased large nodes, and then one chromosome, for whatever reason, due to identity by descent or something else, that are essentially almost identical. And so in this case, there's probably going to be some sequences that are misassigned between haplotypes in this big gray region. In this example, because these nodes are gray, they're going to be unassigned. And this is why I was saying that you want to look at the unassigned bin just in case, because it will indicate that you're missing genes if these sequences don't make it into your haplotypes. This shouldn't happen very frequently now, but I still always warn people to not just ignore the unassigned and take a look at them and make sure you don't have any genes in there or anything, any large sequences in there that couldn't be assigned in the graph. So yeah, the very low heterozygosity causes a problem when we can't, then we have these really long stretches where we can't assign anything, or we might misassign them. And also if you have really low, if you have no heterozygosity, like if you have some parts of the genome that are essentially, some chromosomes are haploid, some aren't, that can cause issues as well. And a common thing that you see in the graph is usually, and one of the most common reasons we don't have a T2T config, but a T2T scaffold usually, are these kind of large repetitive bubbles. And these are kind of things that you could resolve manually after the fact. And we have, if you wanted to spend more time on it, there could be cases where the ONT alignments just weren't strong enough for Verko to automatically resolve them. Or it could be that if you, as a human, you can make decisions that Verko doesn't understand or isn't programmed to do. Like if you could pick one, it has to resolve both sides of a bubble. And if you resolve one side, the other one must go to the other haplotype or the other copy of the repeat, which the algorithm can't do. It has to resolve either all or nothing. And in some cases, you might even want to take a random walk. You're going to slightly mess up the repeat, but it's probably going to be almost identical anyway, which is why it looks like this in the graph. And so you might say you're okay with having a little bit of error in that region if you could fill in that repeat sequence. The other reason that we look at the graphs is sometimes you find interesting biological conclusions or facts from the graph. So this is an example where we have the sequence from chromosome 6, but then it goes into chromosome 3. And so here we have chromosome 3, 2 haplotypes, so each haplotype has about 30-fold coverage, like our standard. But then we go into this bit of chromosome 6 joins us, and now we have this coverage where we have 30x and 60x. So this looks like, the coverage confirms that there's a triploidy here, right? So there's essentially the end of chromosome 6 seems to have a duplication of one of the haplotypes of chromosome 3 in this sample. So again, this is the assembler pointing out an issue with your sample that you can go back and track down and see what's going on. But in this case, it looks like you've had a loss of part of chromosome 6 and a fusion to one of the chromosome 3 haplotypes that's been copied onto the end of chromosome 6 and replaced it. And so now you've got one of these haplotypes of chromosome 3 that's got the standard expected 30x, and then the other haplotype has got double the expected coverage because it's now attached to chromosome 6 and still attached to chromosome 3. And so, we think this could be a real biological case. This is an actual human sample, though we still want to make sure it's actually present in the individual and not just a cell line artifact that arose when the cells were transformed into a cell line. That's kind of always a thing when we see these kind of interesting structures or things in the graph. The first question is always, is it just a cell line artifact, or is it actually something happening in the individual who donated the cells for that cell line? So that's the end of the slides. Let me check the chat for questions. So let me answer the couple of questions. So what do we consider high heterozygosity? Anything over about probably 0.5%, I would consider pretty high heterozygosity. With hi-fi data, with these high accuracy data, we get haplotypes resolved even at the human level of heterozygosity, which is relatively low compared to most other species. And so, essentially anything more diverse than a human, I consider pretty high heterozygosity. We're very human-centric in the National Human Genome Research Institute. And then for the second question, Yes, Verko works well with non-human samples. We've used it on the great apes. We've used it on ruminants. We've used it on some plants, though most of those plants have been very homozygous. So these were like the agricultural crops that are essentially almost identical between their haplotypes. And it works well with that. Most of our testing is done in the human space just because that's what we have the most of the data sets access to for the right data types. But nothing in Verko is human specific. All the algorithm and all the decisions are kind of, they do assume relatively uniform coverage. So if you tried it on a metagenomic sample, it wouldn't work very well. But there's nothing in the algorithm inherently that makes it not applicable to human. That said, it can get tripped up by things that we just didn't expect and we've never seen before. So kind of the things to look for are what I've been talking about. You look at the graph, you see if there's regions of the graph that look kind of off like that, like there's missing telomeres, there's lots of breaks, there's coverage that doesn't make sense. There's lots of things in the unassigned bin, which indicates that there was some kind of mistake in how we were trying to phase the genome. Things like that is what I would say you want to look out for. And like for example, in the chicken genome, and most of the birds have these microchromosomes which don't sequence very well with hi-fi data. So when you look at those graphs, you have a lot of breaks because there's a lot of data, there's a lot of missing sequence data. And so those kind of things can come up in samples that are not human, that are more prevalent in terms of sequencing biases or other issues like that.
I've got a question, if that's all right. So I'm interested in how chromosomal abnormalities would look in these sort of plots. So, like, have you actually managed to find an example of a confirmed abnormality in?
I mean, we've had ones, well, confirmed, you mean an actual individual patient? No.
Yes, yeah,
Um,
someone that previously didn't know of this
um,
difference.
no, we have not. Mostly because we haven't done a lot of patient samples. We've mostly been sequencing the cell lines.
Yeah.
We have found several kind of abnormalities like the one I showed with the 3, 6. Then we've also seen where there's like balanced like swapping of the ends of chromosomes, like chromosome 16 and 17 in a sample in the HBRC. And then there's other kind of large structural variations that we've seen. We want to go back and look at these. So one thing we've done is if we found some of these, we've found a way to type them with short reads, and then we could go to something like the large biobanks and check if we see that in those samples, because those aren't cell line based, right?
Yeah.
And so we do that for some of the regions in the acrocentrics where we think there's like a copy number variable region which has expanded. We can copy count it using short reads, and then we do see in the biobank that this copy number does vary, and it agrees with what we see in the assemblies. In the individual cases, I think what we want to do is kind of lab validation, karyotyping and other ones like that. Like the chromosome 3,6 looks big enough that we should be able to do a karyotype and see it. But I think it'll be interesting to track these down and find some of these.
Yeah, yeah. And how would you imagine something like Klinefelter's or Turner's would look after going through this? Would they, especially with Turner's, because obviously you're getting 2 from the mother, would it be quite a straightforward looking plot where you're just going to see a lot of 60% or 60 times coverage?
I'm not familiar. So what happens in that case?
Turner's is triple X.
I see. Okay. Yeah, so I think you would just have, well, it would depend if you would have non-diploid structure, I would guess, depending on how much variation there is. Is it just, if it's a perfect duplication, you would see kind of what I showed in one case where you have like twice the coverage on one haplotype and half on the other. Apparently doing this makes it have balloons. Yeah, so I would expect you'd see some kind of coverage abnormality there. And the assembler will likely resolve it worse. So like I said, it kind of does assume that at least within a chromosome, the coverage should be about the same. And so when you see these nodes where like half of them are double the other, it makes the ONT resolution not trust the nodes as much. And so it's going to probably be much less resolved, like in that example I showed on chromosome 19 in one of the samples, because it just doesn't know how to find this trustable unique nodes in that graph when like your coverage is all over the place like that. And it says like, I don't know what unique means here because it's all kind of all over the place, right?
It's jumbled, yeah.
Yeah, and so it ends up being much less simplified, much less resolved, and you'll see like just a lot more nodes there in the graph. So I would expect something to happen there. We've definitely also seen like, some cases like large aberrations like Robertsonians come out in the graph very clearly, where you see the same kind of rDNA structure that I showed, but then one of the haplotypes just goes up to it and jumps away and just goes into the other chromosome and just skips that. And it's very clear that they're just skipping the rDNA and go somewhere else. So, I have been happy to see the level to which we can actually see from these graphs, like these kind of expected aberrations. And we've also seen things that people have sequenced cell lines that have known ploidy artifacts and other things. And again, you see that in the graph as well.
Very cool.
Yeah.
Now, how long has Verko been in development now?
I mean, we really started developing it after the T2T project, which was in the 2020 or so late, so probably we started developing it in 2021, 22.
I mean, even in just that space of time, technology has, changed quite substantially. So it must be an interesting task in keeping up with how sequencing technologies especially have changed.
Yeah, I mean, it's been pretty stable though, because we've had this requirement for one, we call it high accuracy, and then ultra long. And so, yeah, the actual thing we use for that accuracy has been changing because we've been using more duplex and more hero-corrected data. But it's still kind of a similar, we haven't really done that much work in the assembler to integrate it. It just kind of is a drop-in. But yeah, really having that high accuracy data as one input has been a key, though, as ONT keeps getting better, there's a question of like, well, are we gonna ever need it at all? Maybe we just don't need the high accuracy data. We can just use uncorrected ONT data directly and not worry about any of this other stuff. And I think that's possible as we keep getting the ONT data keeps getting better. It'll be interesting to see in the next couple of years.
Yeah, definitely.
So I was going to do the quick manual, how you essentially tell the assembler you want to use a different path. And that was going to be the end, but I'm happy to instead answer other questions if people have other questions. I guess then I'll go back to hopefully people haven't timed out. Let's see. Okay, right. So going back to our pod here. So this is an example. So this is something where you've looked at the graph, and let's say you decided you don't like how the assembler has phased this, right? In this case, it's kind of hard to argue with what the assembler has done here, because if you look at these two nodes that are gray, even though they're gray, so we couldn't color them, you have to connect this node over here, because if you connect UTIG 4.1 here to UTIG 4.0, then 4.2 ends up having no neighbor anymore, right? Because its only neighbor is 4.0. So in this example, it's a very, changing what the assembler did here is obviously not something we'd want to do in real life, but just as an example or tutorial, so you know how you would do it. The same kind of strategy is useful if you, let's say there was a repeat or something more complicated, and you wanted to pick a traversal through it. This is also a way for you to tell the assembler, okay, here's how you traverse this repeat. And so the way you do that is you can change the paths that the assembler uses. So we can take these paths that the assembler built, and so we can then, so here's the paths we had, right? And so what I was saying is, let's say we want to switch one of these nodes. So rather than starting this utig 4.1 going to 4.3, let's say utig 4.1, we want to go to 4.0, right? So we would change this and say this is utig 4.1 now. And then we could let the path use it twice, or we could just get rid of this utig 4.3 and just leave this as just a path that has just these two nodes and not, and we can add like unused utig 4.2 and say, yeah, just leave it alone. Right. So you can edit this file as you want, and write arbitrary paths. So these paths follow the GAF format, which I think I linked to from the tutorial page, but this is kind of part of GFA, which defines the path where it says greater than this means follow this path in the forward strand, greater than this, follow this in the forward strand. So this goes reverse. So what we've essentially said is you want to take this node in reverse, this node in forward, and this last blue node in the forward strand. And so we edit this file, and then we can just run Verko again. But this time we're going to tell it, okay, so the same input data. Here's, again, the D is where you want it to write its output. But now, rather than running it before, we say the paths, we use our updated file. And then we point it to the old assembly. So it says, take the graph from, what this is essentially saying is take the graph from this assembly that we already generated. But rather than using the path you generated automatically, use this file I gave you automatically. And now we can find out if I made any typos, which I did, where it now gives an error and it says that it can't figure out how to parse my path. So if you make a typo, it'll tell you that you've essentially made an invalid path here, where it can't figure out how, where I've made too few columns in this one entry. So as a result, it didn't know what to do with this line that was missing an entry. So I could put this in haplotype 2, let's say. Right. Oh, yeah. Oh. All right, let me just try it again and do it a simpler version, where we're just going to say we're going to use this one and we're going to get rid of this one because I don't want to. So now, now I have to remove and try it again. Yeah. And so it'll just run the steps to generate. So let me see if I can do the full updated path now that I got it to run. So I'm going to say do unused. Okay, so I wanted to put that node into haplotype 2 because I decided to switch UTIC42 and UTIC41's haplotype. So I put UTIC41 into haplotype 1. I've erased UTIC42 in the path. If I put it here, it will complain that there's no edge between UTIC42 and UTIC43, which is true because if we look at this graph, UTIC42 has no edge to UTIC43. You can see the only edge from it is here. And so, but I still want to put it into that haplotype. So now, hopefully I did it correctly. Yeah. And so what it's doing essentially is just generating a consensus for this new path. So you can edit them arbitrarily, like you saw. If you make a mistake in the editing, it will tell you what it's having a problem with. So like in my case, I had not put in 3 columns in that entry. They need to be tab separated. And so as a result, it didn't it couldn't parse the lines. And so, there's a lot more you can do kind of with these graphs and resolving them. And so we did a tutorial earlier this year where we kind of went into the more advanced assembly. So here I was kind of doing the intro to assembly, but kind of more the advanced assembly and how you actually resolve, and some examples of how you look at the graph and you resolve some of these tangles. There's a link to a training GitHub, which has a Docker image, and again, has a list of commands that you can follow to actually resolve some of these tangles and build a new path that resolves the tangles yourself, as kind of an example as well. And that goes into the more details of like what you look for when you're trying to resolve the tangles and things like that. So yeah, that's all I wanted to show for today. And I want to, the way we think about Verko kind of, you can run it as an assembler and it'll give you good assemblies, but it's also kind of a very useful tool where you can also run parts of it and use that and modify it and kind of work with it to finish your assembly. So our other goal with this was to also make it easier for us to finish Genome. So this is why it has this feature where you can change how you change. You can update its graph, tell it how to traverse the graph very easily and plug in the outputs from that and let it finish that result rather than having to rerun an assembly again, which if people have run assemblies multiple times, you know that it's hard to like then merge them or what do you do with them? This essentially lets you tell the assembler, no, do it like this, and know that you're not going to change anything else except the final sequence that you output. So that's all that I have for today.
So in a sense, you could use this as an alternative to something like pretext then, in a sense.
Yes, though it's not, it's not going to have all the high C,
No.
right? Pretext is doing a different kind of validation bit,
Yeah.
but you could certainly, it could be a part of the curation. And this is kind of how we do curation on our T2T projects where, we look at the graphs, we see, some regions it didn't phase or it didn't resolve a repeat, and we can resolve it manually, or we don't care, and we're okay if it's slightly wrong in that case. Right, and then we tell it like, do this. And then that lets us generate the final path. So we definitely do use it that way.
Okay, interesting.
And it's kind of graph-based curation, I guess.
Yeah.
Yep. And that's what the other tutorial goes through, is how we do that kind of graph-based curation. Yeah. So if there's no other questions, I didn't have anything else. I wanted to save some time to make sure that if people interrupted me or asked questions, but everyone's been very quiet. So we finished early.
Yeah, it's oddly quiet. Yeah.
Yeah, the Discord channel has also been very quiet. Everyone just fell asleep.
Yeah. No, I'm sure not. No, it was a fantastic talk. Thank you very much.
Yep. Okay.
Yeah. If no one else has any questions, then thank you for coming, and thank you, Sergey, for giving the talk. It's been fantastic. And I will see the rest of you some other time during BGA.
Yeah, thanks everyone for coming.